Welcome, ladies and gentlemen. First of all, I would like to thank all the audience for attending our forum. Our topic for today is induced pluripotent stem cell, which are known as iPSCs under tissue engineering. Before we go further, let me introduce our three panel for today. Mr. Juliana as a scientist. She has a bachelor degree in biochemistry and microbiology from University Putra, Malaysia, a master in biotechnology and a PhD in science communication from the University of Malaya. Second panel is Mr. Tay Wei Xiang, who is a researcher in the field of regenerative medicine. Moving on to the last panel is scientist abroad, Ms. Carol. She graduated from Monash University and trained in internal medicine and infection disease at the Monash Medical Center. Welcome to our forum. Hello, I am Di Cheng Wen as the moderator for today. Without wasting our time, I will begin our forum with the first question. What is the background of this advanced gene technology? May I have your opinion on this question, Ms. Juliana?
Okay, thank you to Ms. Lee for inviting me to this forum. From my perspective, induced pluripotent stem cells, or we call it iPSC, actually are derived from our adult cells such as our skin or blood cells that have been induced or reprogrammed back into an embryonic-like pluripotent state that enables the development of an unlimited source of any type of human cell needed for therapeutic purposes, such as disease modeling, regenerative medicine, and drug discovery. And here's some history about the iPSC technology, in which where it discovered in 2006 by researcher named Shinya Yamanaka in his lab in Kyoto, Japan, in which his group found that by introducing just a few genes, which are OCT34, SOX2, KLF4, and CMYC, into mouse somatic cell, which is fibroblast by retroviral vectors, and culturing the cells for a few weeks, and then the cells could be reprogrammed and or induced to a pluripotent state resembling that of embryonic stem cells, in which it can be differentiated into cells of various types in the body. And his team first reported his success with mouse induced pluripotent stem cells in 2006, and for human induced pluripotent stem cells in 2007. And that is the background of induced pluripotent stem cells.
Thank you for answering this question, Ms. Juliana. Do you have any idea on the example of research or program for induced pluripotent stem cell?
Okay, thank you, Ms. Lee, for the next question. So, I choose one example of induced pluripotent stem cells from disease modeling, which is Parkinson's disease. And I think everyone is familiar with Parkinson's disease, right?
Is Parkinson's disease the disease where one will have difficulty in walking and talking?
Yeah, that's right. And actually, Parkinson's disease is a brain disorder that causes unintended or uncontrollable movement, such as shaking, stiffness, and difficulty with balance and coordination. And for your information, Parkinson's disease affects approximately 1% of individuals over age 60. And iPSC are the tool used to model Parkinson's disease, in which fibroblasts from patients carrying the pathogenic mutation that lead to the Parkinson's disease have been reprogrammed into induced pluripotent stem cell, which can subsequently be differentiated into important cell types. Given the typical loss of dopaminergic neurons in the substantia nigra pars compacta of Parkinson's disease patients, midbrain dopaminergic neurons grown from the induced pluripotent stem cells have been created to study pathogenic pathway in this crucial cell type as the way of modeling Parkinson's disease.
All right, thank you. Basically, from Miss Juliana, Parkinson's disease usually happens among all people like over 60 years old. They will have the conditions like shaking, stiffness, and difficulty with balance and coordination. Next, to make our forum more interesting, Mr. Tay, as a researcher, what do you think about the status of this technology in Malaysia? Is it available or under research and clinical trials?
First of all, I want to thank Ms. Lee for inviting to this forum. Induced pluripotent stem cell (iPSC) is considered as a type of regenerative medicine technology. In Malaysia, the only approved stem cell therapy by Minister of Health currently are bone marrow and peripheral blood stem cell transplantation. The application of stem cell for other indication is still in the early research stage. Talking about research, in 2012, a research group in University Tunku Abdul Rahman, UTAR, under Professor Chong, was the first to generate iPSC in Malaysia. With in vitro reprogramming procedures, adult somatic cells were reverse developed into special stem cell known as the iPSC. Using virus-free and serum-free technique, the research group was able to establish clinical-grade GMP-compliant normal iPSC cell line from normal skin cell and from hematopoietic stem cells.
Thank you for the explanation, Mr. Tay. It seems that iPSCs have a lot of potential to develop. May I know what is the potential do you see for iPSCs in Malaysia?
Currently, there are numerous ongoing research and clinical trials carried out and have shown the possibility of clinical application of regenerative medicine technology in Malaysia. With many unique properties, iPSCs have many great potential to support the research and development, such as developing human tissue and human organ when given the right conditions.
Sounds great. Thank you for your explanation. I would like to continue with the next question. Do you have any opinions on how this technology can be applied to the local problems or situation in Malaysia?
Thank you, Ms. Lee. May I ask you a question? Do you know what is the major health problem currently in Malaysia?
I guess it's diabetes or heart attack.
Well, you are correct. One of the major health problems in Malaysia is coronary heart disease. According to the World Health Organization report, coronary heart disease is Malaysia's leading killer, and about 23% of all Malaysian deaths are caused by it. There is a research that shows pluripotent stem cell-derived cardiomyocyte can form beating human heart muscle cells that both release the necessary signal and also replace the muscle loss caused by heart attack. And recently, pluripotent stem cell-derived intervention was used in clinical trials. Patches of human heart muscle cells derived from stem cell were transplanted onto the surface of a failing heart. However, it is still in the early stage of development. Therefore, more clinical trials and research need to be carried out before it can be applied to local situations.
All right. Thank you to our panel, Ms. Juliana and Mr. Tay. Let us welcome our third panel, Ms. Carol, who stay in the United States by watching the recorded video provided by her. Let me play the video clips. Hi everyone, Professor Lee here. Thank you, Ms. Lee Cheng-Wen, for the inviting, and I'm so sorry that I can't attend this online forum because I'm still busy with my medical research. Oh yeah, regarding the question about ethical and health issue, there are a few issues that can be raised, such as privacy and volunteer participation. For privacy, iPSCs derived from any individual, like any cell, will inherently contain a vast amount of confidential personal information like DNA. If used carelessly, it will violate law, morality, and individual privacy. Even if the initial cell donor is no longer alive, the iPSCs still contain information about his or her close relative, potentially raising ethical and legal concern about individual privacy. On the other hand, iPSCs may require ongoing access to information about the donor health status, which require knowledge of the donor name and address. For your information, it is mandatory to inform and obtain voluntary consents from participants whenever humans or their cells and tissue are to be used in research projects. In general, one of the involved researchers who is closely associated with all the research content should explain the essence and the purpose of the research to each participant. In cases where the patient is unable to make a decision, to the patient's custodian, and then discuss the potential side effect of the treatment with the recipients if the cells are intended to be used for therapeutic purpose. Patients should be aware of their right, the duration of the study, the circumstances of their withdrawal from study, the risks, and the number of participants. For the health issue, iPSCs can accumulate chromosomal abnormality, genetic instability, copy number variance, and loss of heterozygosity over periods of time in vitro culture. That's all from me. Thank you. We have come to the end of our forum. There are a lot of questions and answers that I want to share with our audience since time is running out. I will conclude in our forum that iPSCs have opened a new avenue for stem cell research and unique opportunities in the pharmaceutical industrial and clinical practice. However, reprogramming technology have its own ethical social issue, all of which must be carefully considered. A lot of input we have received, hoping that our audience can gain this knowledge and implement it. I would like to thank all the panelists for attending this forum. Thank you.